So, welcome back. Hope everyone's doing okay. I wanted to give us kind of a chance to reset if anybody's been joining us remotely and maybe now you feel a little more seen than you have before. In the classroom we had previously, I was, I couldn't really see anybody. You were projected onto a television at the back of the screen, and now that we're maybe all sitting in front of computers, get a little better chance to weigh in. So if you haven't been able to join or share or anything like that, now's a chance. Now, a few minutes from now, any time during the process, you can chime in, share anything. So obviously we got a short week this week. This is our first time being in a different format. We're in between an exam and the material for the last exam, so it's kind of a little bit of a break point here. So it's a funny thing to have on the announcements. So a few things about announcements. I will be updating grades. I thought I'd be farther along than I have, but I've got a little bit of a backlog for lab grades, lecture grades. I got the assignment 6 graded. People didn't do as well on that as I thought, and I did, somewhere in that process, I think the day it was due, I did basically a walkthrough of the assignment 6. So if you wanted to find that video and review what we covered for assignment 6, my intention with that was that people could go back and fix anything if they hadn't gotten the right answers the first time around. And then I didn't get a chance to really review those before the exam, so I'm guessing maybe the exam being in a different format, mostly multiple choice, people might have overcome if they were not clear about some of the correct answers on assignment 6. They might have gone ahead and done okay on the exam. The exam I haven't graded. There's a couple on there I have to grade and just maybe double check. So I'll be doing that hopefully soon. So you should get a pretty rounded out version of your grade soon. I'm also hoping or planning to incorporate the lab grade. So your total grade in the course, it's 4 credits and they all go towards 1 letter grade. And that's a combination of your lecture and lab grade. People typically do much better in lab. It's just if you do the assignments and you tick all the boxes in lab, you can get in the 90% for a lot of people. And that's going to come in as like 1/3 of your total course grade. And once I do that, that'll be a little bit more realistic version of your grade. Right about now is a good time to maybe take stock of your grade once I get those posted. And if anything is, on the verge of where you want it to be something different or you like it where it is, this is maybe a good time to know about your grade. So I will get all that up. That's on my agenda. This little note about the final exam. We've been asked to do these in person or, like live at 7:45 in the morning on Thursday. I just the whole semester, and I think we're basically being asked by the university to proceed being flexible. So this is a nice semester. My exams always fall at 7:45 in the morning, but this year you get a break. And you can take it anytime on that calendar day. So I'll do the final exam just like I've done other exams, where you'll get a 2-hour block to do it. You'll get practice questions ahead of time. The format will be very similar. And it's not really cumulative. So I think it's worthwhile if you can recall all the material we've covered up to this point, but I don't think it's worthwhile for me to dig back on something we covered in the 4th week and pull out some minutia for you on that. So I'm hoping that this course is memorable and you go home with a lot of exciting new information about genetics in your pocket. But as far as what's going to be on the final exam, that'll be the material basically we cover between now and the last class. So we have 5 scheduled classes, and that'll be the material that'll move forward onto the final exam. So, any questions up to this point? All right. If I can see you any better. No? All right. You can see each other pretty well, I imagine. Maybe not. I see mostly my screen and little people in the corner. So, nothing? How's the new living situation? Back at home? Some people have changed their living situation. Some people are, I don't know, hanging out in the dormitory right now. All right, somebody say something. Chapter. Any exciting genetics discoveries over the weekend? All right. Well, don't tell me later you didn't have a chance to weigh in on anything. All right. As far as the schedule for what we're going to cover from here out, I have more topics on the syllabus than we'll have time to cover. So I went ahead and I just went with the topic that was scheduled for happening after mutation, which is biotechnology. Kind of a nice, I think maybe a nice little palate cleanser topic. It might get you interested about something we can talk about. And then after that, we'll do gene expression. And gene expression usually takes us a little while to cover, so that might be most of what we cover for the final exam. We'll start that next week after Thanksgiving. What was I going to say? I don't remember. Hard talking to a blank screen. Okay. So, biotechnology is kind of a new, I suppose it's not very new at this point, but it's a field of study that basically builds off of what we know about genetics and heads into new territory. So it's like bioengineering. If you're looking for interesting careers, this is a topic that is really a booming industry now. So taking what we know about genetics and using that to make change in organisms or understand even greater about phenotypes and their causes or inheritance of diseases or something like that. And you're positioned to go places once you've understood genetics. If you're interested in this topic, there's a molecular biology course at Whitewater. That's an upper level course, Biology 363. And there's also the Biotechnology Lab Methods course. For lab, the last 2 weeks, those will be entirely remote, of course, and those are, we'll cover the topic of population genetics in the lab, the last 2 labs. So basically, instead of doing the exercise, you'll be watching videos of the exercise and answering questions similar to what you would have done in person, which is just as well because in the first lab of the population genetics series, we have you grabbing spit from your cheeks, and that's probably not a great thing to do if people have to wear masks into a lab. All right, so moving forward. So biotechnology is a really big field and encompasses all kinds of things basically related to kind of manipulating organisms based on what we understand about them, and a lot of times that comes down to genetics. So we have lots of exciting tools now to make genetic changes to organisms. So some examples here. This is a technology that's been in place for some time now, where researchers can alter the DNA of certain genes, and those genes get expressed and make proteins, and the protein comes along with a fluorescent tag at the end of it. So maybe I'll do a little doodling here. So if we have a normal gene, and let's say we've got our neural motor Start codon, stop codon. And let's just skip the processing step. So we'll go right into, let's say there's no introns here to worry about. Through biotechnology, you can add, keep that on the same side, you can add pieces to a gene, you can edit them. And to edit genes for this green fluorescent protein technology, you basically add a little bit of extra protein coding sequence into the gene region. So it's like added in there to an existing gene. And when the protein gets made, there's the normal amino acid sequence. So normal protein plus the GFP region, this green fluorescent protein, which is an exciting protein. Has an interesting look to it, has this kind of barrel shape. And again, this shape of the protein relates to its function. And it happens to fluoresce under, I think it's ultraviolet light. And so this is basically an original green fluorescent color. And through, again, a little bit different manipulation, you can alter even the green fluorescent protein and turn it into a red fluorescent protein or a blue fluorescent protein. So this was originally taken from a jellyfish that happens to be fluorescent. And they isolated the protein that makes the fluorescence, and they said, okay, we're going to take that protein and actually smush that fluorescent protein onto existing proteins in the laboratory. And so the proteins found in muscle fibers get an attachment in the lab that makes them red. And I don't know my animal anatomy very well, but this other green bit here is highlighted with green fluorescence. There's some, look like maybe nerve connections here with the blue fluorescent protein. You can basically paint the parts of an organism using these fluorescent additives onto the proteins. So this is something right now that's done in the lab. They don't do this and then release these multicolor fluorescent mice into the world. It's done to, in this case, maybe just for a nice picture, but you can also do this if you have a protein that you don't really know where it is in the organism. You can tag it with this fluorescent protein, and the fluorescence will show you everywhere that that protein is found. So they do this to find out where proteins exist inside of single cells or in organs of bigger organisms. So this is a new technology. To do that, as I showed you on the notes here, to be able to get this fluorescent color onto a protein of your choice, then you have to be able to alter the DNA of the organism. So this is a lab mouse with altered DNA that causes it to make proteins that have this fluorescent component to them. Have you heard of GFP before? Maybe. I can see like two of you at a time. Most people are without camera today. So the technology, the form of technology here where we're combining DNA from different organisms, that's called recombinant DNA technology. We saw recombinant before in the context of crossing over. So recombinant is not a, I don't know, I don't know if it's a very specific term. It basically refers to new combinations. So in crossing over, we saw new combinations of alleles relative to the parent. In this situation, we're seeing really new combinations of genes that were not necessarily even available to the starting organism. So, in the laboratory, two organisms that never would meet or have any chance of exchanging DNA, a mouse and a jellyfish, and take bits of one and put it in the other and see what happens. And again, this really depends on knowing a lot about how genes work. So being able to put it in there, understanding the coding sequence and how it translates, understanding the reading frame for how the codons would get translated, understanding important bits about promoters and gene expression, and so on. So it takes a lot of upfront knowledge to be able to do something like this. So maybe you've encountered some situations where this is possible. This is a tool we have. This one example of green fluorescent protein is a tool that can be used to zero in on protein location in an organism. But the possibilities are, I'd say, downright scary, right? So if we understand the DNA of organisms and what it does, we can affect the DNA of existing organisms. We can affect the combined DNA from different organisms and so on. So who knows what could happen. There's a news story this week where they were, maybe it was last week, growing bigger brains in primates. Anybody see that story? So non-human primates, they did a little bit of altering to their genes and they got bigger brains. So it's scary what we can do with organisms. I think that was the plot of Planet of the Apes, right? Yes, they're doing it now. Okay, so lots of opportunities. One of the common organisms that gets influenced with biotechnology, so you could do it sort of most simply using bacteria. And this technology goes way back. Bacteria are simple organisms, so single cells, small genomes, bacterial transcription translation, so not the complex RNA modification things we saw in central dogma. So in bacteria, just like DNA of a gene becomes RNA of a gene, a little bit easier to get that to happen. And if you're just looking to make a protein, you can enlist bacterial cells to make the protein for you. So really take whatever protein you're interested in. We know how to translate genetic code into proteins. Basically, you can just throw that code into bacterial cells and they will make the protein, and then you can purify the protein that comes out of that. I'll show you some examples of how that's done. And again, pretty common. If you just need an enzyme or a protein, bacteria is probably your source for those. That would still be outside of the organism. So let's say you make an enzyme that people need. Making that in bacteria means you can just give the person an enzyme and not necessarily train their cells to make. A little more complicated would be to make changes to the cells of an organism. So multicellular organisms are a little more tricky. We have lots and lots of cells, and they've all got their own genetics, and so making changes, it's not easy to just change all of the cells of my organism. I'll show you some tactics that could be used for that. So some more examples. This is one that's kind of notorious, and it's been, or I don't know, it's been known about for a long time. And I think it's maybe just beginning to be implemented. There's been some kind of some regulatory struggles with this one. This is an example. They call it golden rice. And it is a biotechnology option to take rice, which is typically pretty low in nutrients. Rice is good for an energy source, carbohydrate source, but it's not like a complete nutrient diet. But most of the world, more than half of the world, lives on a whole bunch of rice. So big parts of Asia, rice is a major part of their diet. And parts of Asia, as other parts of the world, would have issues with maybe nutrition or things like that. And so scientists came up with this option to genetically engineer rice to make, in this case, they're making beta carotene. So it's like carbohydrate plus vitamin all in one. So really, they wouldn't have to change anything about what they do. They can continue to eat rice, but if they're eating this modified rice, they're getting a vitamin as part of their daily food staple, and it's cool colors. Double interesting, I think, on this is that the gene that they use to do this comes from daffodils. So lots of plants are able to make beta carotene, but in this case, they, for whatever reason, that was their source. And they say, we're going to take daffodils and take their gene for making beta carotene and make it so that the rice plants would be able to make their own beta carotene, but they're not naturally socking that into the grains of their rice, the food part of the rice plant. And so through engineering, you can get the rice plants to put into the starchy part of a rice seed, put in this beta carotene. So some interesting options available to people. Another one here, if anybody takes supplementary insulin, where does your insulin come from? What is insulin? Insulin is an enzyme. So here over here, insulin, a protein that regulates glucose uptake. So people with diabetes have an extra challenge of regulating their blood glucose. So different types of diabetes. Some people have a situation where they're often too much glucose in their blood, sugar, bloodstream. Some people, other kinds of diabetes, too little glucose in their bloodstream. And this one, type 1 diabetes, can be regulated using insulin shots. So where do you get an enzyme from? Well, we could train the cells of this person to make their own insulin. That's, as I said, a lot more challenging. Another option would be to get bacteria to make this for them. So I kind of talked about this in general terms. Maybe we could think about a little more specific, the process of getting this insulin into a person. All right. So let's say we had, we'll just make it simple and have two people. So some people would have the to restart my notes once in a while. It doesn't like to cooperate. Let's do that again. Notepad. All right, let's say we have two kinds of people. One person can make the functional version of the insulin molecule. Somebody else would have the non-functional version of the insulin molecule. So the person who can make the functional insulin has the normal or the functional version of the insulin gene. Somebody who is not making this functional insulin would have a mutated version. So the goal is really to get the information that is present here and offer that to the person who has the non-functional insulin. And again, we can't really give the gene to this person. Why not? Does that make sense? So if you have a person and even if you targeted the location in this person that's making the insulin molecules, the insulin enzyme, it's still pretty challenging to alter the genetics of cells in a living person. Probably prohibitively so at this point. I think we're probably close to maybe being able to do that. So basically too many and too complicated. To change the DNA of the cells in this person. So rather than try to alter the genetics of a living person, a living adult, instead we're going to, or people have come up with a strategy more to give the enzyme directly to that person. So we'll see a little bit about this process. Basically, you take the gene for the protein you want to make, and you can give it to a bacterial cell. This is a process that takes advantage of basically bacteria are expendable. You can try to do this on millions of bacterial cells all at once. It's not 100% efficient, but you might see this having success 1 out of 1,000,000 times, and that's still pretty good for bacterial cells. So at some rate, you're able to put in the gene that you're interested in, add it to a bacterial cell, and we'll call this an artificial chromosome with the gene. With the gene. So this altered version of the bacterial cell can make the insulin protein, and then you can basically chemically purify or enrich for the insulin protein, and then give that as really just, that's the drug. So when people need an insulin shot, they're adding this enzyme to their body, it gets incorporated into their bloodstream, and they can regulate the presence of the abundance of glucose in the blood sugar. So this process, if you want to do it, if you wanted to start from scratch and be able to do this, you'd have to understand the genes. You need to know what gene makes insulin. Sometimes it's more than one gene involved. Have to take the instructions for that. So coding region, start codon to stop codon. Take that DNA out of the person, really kind of make a copy of that DNA from the person, and then insert that into bacterial cells and give it back to a person. All right, good so far? Okay, so insulin, we got the DNA instructions from a person who's able to make insulin, or we don't really need a person to start with necessarily. We just need a source of DNA, I guess, part of a person. And then bacteria can really stand in as this organism that can take DNA instructions and make proteins. And we'll kind of talk about this process. So putting DNA into that organism and expressing the gene. So turning that gene instructions into protein. All right, so this topic really gets into territory that you're going to cover more carefully in biotechnology lab methods, if that's a course you take for your major. If you're interested in that, you can certainly take the course as well. It's a course that's chiefly devoted to lab experiences and relating to this kind of technology. So I'll tell you about it, and if you want to do more as part of that course, you can definitely take that course in the future. So my simplified version is to talk about this process as though you were using the tools on your I don't know, document editor, right? Cut, copy, and paste. We're all familiar with those by now. And that's kind of the process for moving DNA into a new organism. So it's kind of a little bit in a different order. Copy, then cut, then paste. So we need to make copies of the gene or DNA, whatever we're interested in. And then this process on the illustration shows cutting. So making a cut into a double-stranded piece of DNA and then allowing that to be added to, as we'll see, this circular piece of DNA is an important way to keep DNA intact as part of a bacterial cell. So this one illustration here shows basically the cutting and the pasting part, and then we'll talk about the copying as well. So I have this term here, vector. Vector is just a term that refers to something that carries something else. So a vector is this piece of DNA, this circle of DNA that carries the gene with it. So not all of this is the gene, the orange part. Other parts in gray will be important, as I'll show you. But the fact that it's a circle and has certain components in the DNA of that circle allows it to be put into the bacterial cell and maintained in the bacterial cell, not just destroyed. So bacteria, they like their DNA in circles. They don't get chewed up because there's no end to chew on. And so people have figured out that they can add this circle of DNA to a bacterial cell and it'll be maintained there. And then this other term, plasmid, that's a term that refers to this circle of DNA. All things that will probably make more sense later if you ever do something like this. So as part of this topic, the copy process is a technology that's been around for a while now, and it's something that we'll do. You'll get an introduction to this also as part of the lab, so I wasn't sure how much to do with this, but I thought it probably wouldn't hurt to have it more than once. This is a process called polymerase chain reaction, or PCR. Heard of it? Heard of it. All right. This is, I'll try to incorporate some of this into the prep for the lab. This is one of the technologies that's being used to determine if people have coronavirus. So maybe you've heard about the PCR tests, hopefully. So one of the testing technologies is you take a sample from a person, they do it up the nose where the virus is most likely to be. If somebody has the virus, that's more likely to have evidence of the virus in that place way up in your nose. And then they take that and they basically, they may have to purify the DNA, or they may have technology they can just kind of skip the purification step. And they use this process, this PCR process, to enrich for DNA that is unique to the virus. So they have what are called primers here that match the piece of DNA that's going to be copied. And if they match in the right place, that can tell you that you have a piece of DNA. Or actually, you can work this on RNA as well for viruses. Being able to make a product requires having the unique starting place, and so having that starting place is basically evidence that you have coronavirus. So this is doing this process is one of the ways that people can take a sample of fluids from your body and find out if there's viral RNA in there. All right, so the basic process here is to take a double-stranded DNA starting point and add these primers to them. Primers are short, like 20 nucleotides long, single-stranded pieces of DNA. And there are forward primers and reverse primers. So these two strands of DNA, they separate out, and the forward primer will attach to one strand and the reverse primer will attach to the other strand. The primers are designed by people, and they're designed to match pieces of DNA and to copy a reasonable distance of DNA. So usually just a few hundred nucleotides get copied during this process. And that happens using a polymerase. And at the end of one of these rounds, there should be going from one double-stranded piece now to double-stranded pieces. And this process can be repeated. That's the chain reaction part. So repeat the process. These double-stranded pieces can also be separated and copied. So your two double-stranded pieces now become four double-stranded pieces. Another round of the process gives you a copy of each of those. So 4 times 2 gets you 8 double-stranded pieces, and so on. So this chain reaction can go from one starting double-stranded molecule, and in a few rounds of this, because of exponential increase, you can end up with 1 billion copies. All right, so I mentioned the primers, forward primer, reverse primer. The copying here, the increase of size for this red piece, that's responsible, or that's due to the action of a DNA polymerase. So an enzyme that would see this single-stranded gray molecule and make a copy of it, a complementary strand for that gray piece. That's the job of the DNA polymerase. Another ingredient here are the building blocks for new DNA, known as dNTPs. The D is deoxyribo, so that would be the backbone for DNA. N here is just nucleotides, so deoxyribonucleotide triphosphates. So that means A, G, C, and T building blocks, part of the ingredients you add for your PCR. And then, of course, the starting DNA or the template DNA. So you can't make a copy without starting with something to copy. So having the right ingredients is important, and then also going through these temperature cycles is part of the process. So it's sort of like an artificial version of what naturally copies DNA in cells. So in your cells, when it's time to copy the DNA, the enzymes get involved, they separate out your DNA into single strands and copy the single strands using a polymerase. We didn't cover that much in this class because I figured you had it before, and it's not, I don't know, it's not terribly exciting from a genetic variation standpoint. All right, so instead of the enzymes that separate out double-stranded DNA into single-stranded DNA, you can actually take advantage of temperature. So at high temperatures, the two strands of DNA will come apart. They're held together by hydrogen bonds, and high temperatures cause those to relax a little bit. And so you get your double strands become single strands. And then at a lower temperature, this so-called annealing temperature, the primers get in there. So high temperature, the DNA separates. At lower temperature, allows the DNA to become double-stranded again. And because of the number of primers there's lots of primers, and they kind of get in there first, and they attach where they are complementary. And then at a little bit higher temperature, is the process of extending. So that's where the polymerase gets in there and makes the copy. So to do this in the lab, you just run this through cycles. There are machines called thermal cyclers that raise and lower the temperature. That's what they do. They're like a fancy, rapidly temperature changing oven. And at the end of every cycle, you should have twice as much DNA as you started with. Knew that was going to happen. Sorry, I got to pull this up again. No, that's dangerous. Heading into YouTube here. Ah. I really like that video, and I don't remember where the other one is. I will. I'll try to pull this up for the lab. I'll have a video for everybody as part of the lab. So it's basically a video version of this where the primers attach and extend over different cycles. Questions up to this point? So one of the big advantages when you're doing this polymerase chain reaction, if you remember, from one slide previous, this temperature that denatures or separates out those double strands of DNA, that's a temperature of almost boiling. So 94, 95 degrees Celsius, 100 degrees is boiling. So this is really hot. This is a reaction that happens outside of living organisms. So you take the DNA out of a person and then you run the PCR on them. One of the potential problems is that enzymes and proteins, if you heat them to high temperature, a lot of times they denature, right? So when you cook your egg, it changes the components of the proteins and they don't function anymore. They go from liquid egg to solid egg and they don't go back. That's the process of denaturing. And so if you did that to a normal, like a human DNA polymerase, it would not handle this high temperature and you would destroy the enzyme in the process. So how to get around that? Fortunately, there are organisms that have DNA polymerase that can handle living at really high temperatures. So the DNA polymerase that's used for PCR is called TAC, and that's just basically a condensed version of the scientific name of the bacteria that this was originally sourced from. So again, if you want an enzyme, this is an enzyme that is commercially valuable, and right now they can get laboratory bacteria to do this, to make the TAC polymerase. You can get an ordinary E. coli to make the TAC polymerase by taking the DNA from this Thermus aquaticus bacteria and teaching an E. coli cell how to make this. So it's like biotechnology for the service of biotechnology. So this is an image of a hot spring at Yellowstone. This is where, sort of interesting tidbit, this is where the TAC polymerase was first sourced from. This is a part of the world that has really hot water and organisms that live there. So there are bacteria that can live at really high temperatures. So temperatures around boiling, still not prohibitive for life. Life's pretty amazing. So this is just a little image of the bacteria, the TAC bacteria polymerase. And the natural version of the TAC polymerase, that was really the starting point, and it's still a pretty big industry. So by tinkering with the polymerase, scientists can figure out ways to make it faster or more efficient, or they have extra additives that like protect it from high temperatures and all kinds of stuff. And there are lots of biotechnology companies out there that will sell you variations on TAC polymerase, all with their own like trademarked taglines on there. So people can deliberately mutate the sequence and kind of see what they get, or maybe they can predict what they would get for a more effective, like a more stable polymerase, a more speedy polymerase, whatever you like. No end of the research that goes into this. I think I want to skip over this one. This is something we'll do in lab. It's called gel electrophoresis. I'll get a nice video of that for the lab component. What's our time like? So we talked about copying. Copying is the result of using PCR and that Taq polymerase. The process of cutting, this is one way to cut DNA. There are enzymes called restriction enzymes or restriction endonucleases. And what they do is they recognize a specific DNA sequence on double-stranded DNA and they cut it. So they're like molecular scissors. And if you add this enzyme to DNA, it's going to cut everywhere that recognizes, or that every sequence that it recognizes. So there are different kinds, and they have different names like this. So Hindi3, as it shows over here, is named for a bacteria called Haemophilus influenzae. So the HIN, HIN, H for the genus, IN for the species name. And then D3, there's often lots of different restriction enzymes that could be isolated from the same bacteria. So they get a name. People find out that they're useful based on how they cut. And so based on the unique properties of DNA sequence, you can take an existing DNA sequence and find out where these enzymes will cut. You can alter your DNA sequence to make it so that an enzyme will cut somewhere. There are hundreds of these restriction enzymes, and so you can likely find the kind of sequence that they would cut depending on how you need to cut your DNA. So a couple examples here. Hindi3 is cutting in a way that has overhangs, and this PVU2 is cutting in a way that leaves what we call blunt ends. And the overhangs are kind of useful. So let me memorize that sequence and show you how it works. AAGCTT. AAGCTT. All right, they don't tell me 5' and 3' end. I'm going to take a stab and say that this might be the way it is. So that's the one strand of DNA. Then it would have a complementary strand. And the way that this one cuts is to cut this TCGA sort of cuts here, and then it cuts there. So this ends up being a pretty useful way for DNA to be cut because it leaves what we call sticky ends. And they are sticky. No. Change that and say it's sticky for a complementary strand. So if you have a sequence here, I'll just make up some sequence to the side of this. This end of the double-stranded DNA molecule, as it exists, it's got these single-stranded nucleotides just hanging out there. And single-stranded nucleotides would love to pair with other single-stranded nucleotides. So somewhere out there floating around is somebody who's got a A, G, C, D, and that one's good. Did I do that right? Yeah, sure. I'll throw an extra T in there just to be safe. So if this sticky piece encounters this sticky piece, then they will be complementary to each other. So T complementary to A, C to G, G to C, A to T. They would find each other and match up. And so in the lab, you can do this. There we go. I'll go back and fill that in a little bit more. You can do this to cut the DNA from two different pieces and cause them to be sticky to each other. So if you take a red piece here and a blue piece and cut them both, you can cause the blue piece to stick to the red piece. Because after they're cut with that HindD3 enzyme, they're sticky in the same way. That make sense? Let me go back to here. I think this one illustrates it nicely. So if you have let's say you have the circular plasmid and a piece of DNA you want to insert into it, if you cut them both to make these sticky ends, then the insert will be sticky for the plasmid, and you can cause it to attach and add itself into that circle. And then I guess if you want to finish this up, the paste part of the process is an enzyme called DNA ligase. Once these pieces are sticky for each other, they're still not quite complete, and the DNA ligase comes in and seals up the DNA backbone there, which would need to happen for this to be really a permanent addition. All right, so let's zoom out a little bit more and think about how a person might add a gene to a bacterial cell and get it to behave like a gene. So that becomes a little more tricky, right? How do you add coding sequence? So you have the protein coding sequence from your source organism. How do you give it to bacterial cell and get the bacterial cell to know that it's a gene? So also related to what we saw before, how does a cell know where a gene is? And that comes down to the promoter here. There's a little more on here than we need to see. But we see some familiar pieces to this part of the plasmid, the circle of DNA. So it's got a promoter, it's got transcription initiation sequences, so basically part of this that's meant to be another form of the promoter. So the promoter will get the RNA to be made. There's this ribosome binding site. You remember that from bacteria? So ribosome involved in translation. So transcription starts, and then the RNA that gets made also has a code to begin translation. And then these arrows here mean that there's like a sort of a customizable space on this plasmid where you could insert a gene and have it be part of what gets transcribed and then translated. So it's in between the transcription initiation and transcription termination, so between the promoter and the terminator. and it has a code for translation to happen. So you can add in a gene, and this bacterial cell that gets this plasmid will treat it like its own gene and transcribe and translate it, and then you get your protein. So a lot of what's on the left here, we can kind of ignore that for now. Other parts of this that are important are this ORI region. ORI stands for origin of replication. A little note in there. The origin of replication, if you have a bacterial cell and so use the schematic that I did before, where we've got this big circle, which is the bacteria's own circular genome, and then this little circle is the plasmid that we added. Each of those have an origin of replication. That means when the bacterial cell divides, it's going to copy its own genome, but it also takes care to copy the artificial, it's basically like a little artificial chromosome that we add in there. And if the ORI sequence wasn't part of that, this wouldn't get copied. So that's important. Another important part is this antibiotic resistance. And I'll show you what that does. That makes sure that every bacterial cell that we can basically choose among bacterial cells, not all of them would get this plasmid or keep the plasmid, but we're going to make it so that they need the plasmid, otherwise we'll kill them. So bacteria that have this artificial plasmid will be resistant to antibiotics, will expose them to antibiotics, and if they don't have the plasmid, then they would be susceptible, they'd be killed by the antibiotic. All right, I think I basically talked about that. Let me just show you the, skip the text for now. So if we take our plasmid and add it to bacterial cells, and let's say that not all of them got that, or some of them lost it. If the plasmid contains the instructions for being resistant to antibiotic, and we give antibiotic to these cells, that's going to kill the ones that don't have the plasmid. So it's called a selectable marker. The selection in this case is artificial selection for resistance to antibiotic. All right, so I think I want to jump ahead to a little bit more content. I think you might have reached your limit of the amount of DNA you want to talk about today. Okay, so just, I guess, moving on to other organisms, it's possible to make genetic changes. I showed you the altered genetics of the mice limb that had the fluorescent proteins in the muscle fibers. So it's possible to change the genetics of other organisms, multicellular organisms, And one consideration, so as I mentioned, you really can't take an adult and alter the genetics of all of its cells, right? So the cells are individual cases. Changing every one would require tools in a different way than we really have. So when you alter bacterial cells, you're kind of playing a numbers game where it's going to be, like I said, sort of 1 out of a million success. If you try to alter the cells of a mouse and you got 1 out of a million, you might not be very effective for what you're trying to do. But wait, there's a solution. So something like in the laboratory where you have mice, if you target the cells that become a whole new mouse, then you can alter the cells of a whole organism. So if you allow yourself time and generations, you could make changes that affect the whole organism. And there's, I don't know that it's being done in people, but it's certainly talked about, right? So if you find out that you have a child or you have let's say a family disease in your family, genetic disease, and you wanted to do some kind of gene therapy to change what happens for your children, this kind of technology could be possible. So to do genetic manipulation of single cells, egg cells, sperm cells, zygotes, that would be possible to make a genetic change that affects a whole human. It's kind of controversial, and I think it's been done in some places, but not widely yet, although we'll see what the future holds. In the lab, a little bit more, and make changes to organisms in the laboratory. And so here's a case where they are trying to make a change genetically. They can do, this is an example of using PCR to find out whether they made the change they were interested in, and potentially, as it says here, use mice that might have a heterozygous genetic change, and then they can breed in different mice with that heterozygous change and try to make homozygous mice. So that's what's shown here is crossing A by C. A has the gene that we're interested in. C has the gene that we're interested in. And if we cross A and C, then some of their offspring will have a chance of being homozygous. Another option for humans, there's an option to potentially take cells out of a person and sort of grow them up into tissues. So we can do what's called tissue culture to take individual cells and grow them into different kinds of tissues. So if we take what are known as stem cells, they have the ability to turn into other kinds of cells. So if a person needs to grow new skin or a new piece of liver, then the starting stem cell can be genetically altered, put in an environment where it turns into, say, skin cells, and then those skin cells can be given back to the source person. So this is also a technology where you can use your stem cells or stem cells that are compatible for you, ones that your body won't reject from another person, and grow new tissue. So this is promising for nerve damage, or let's say if somebody has genetic deficiencies that are affecting the kidney function or something, you could take, again, sort of normal functioning or properly functioning cells and then sort of add them back. So if a person has a liver that's not making enzyme, they could be provided with some cells that know how to make the enzyme. And this is another promising technology. I don't know how much of this has been done lately. It was exciting when they were first able to do it, and I don't know if there's a whole lot of cloning going on, but cloning is an option. So this is a situation, this is the first cloned organism or multicellular organism here, the sheep. Let's see, first cloned, I don't know, artificially cloned mammal. I don't know what it counts as. But they basically took a zygote, emptied out its native genetic material, added back genetic material from an adult organism. And so basically, if you take a genome, my genome, and add it back to a cell, and make that as a diploid cell, that would have the genetic instructions that are the same as my cells have and turn into another genetic version of me. Creepy. I don't know if there's a lot of interest in this, but certainly good sci-fi and maybe, I don't know, maybe some connections for other types of organisms. But would anybody clone themselves if they could? Yeah, interesting. I don't know. I think having kids is, it's not genetically the same as cloning, but it's a lot of the same stuff. You get to make a new version of yourself, 50% like you. That's pretty good. One of the technologies that's a little bit newer on the scene is this CRISPR-Cas9 technology, and it's fairly recent, and it offers a whole new potential. It's basically enzymes that can go in to the cells of living organisms and target specific genes and make changes. So one way to do that is to simply inactivate those genes. There's a little bit different version here, adding DNA in. And this serves a function that we weren't able to do for a long time, of really making these controlled specific genetic changes to a living multicellular organism. A lot of the tools we've had require like carefully taking care of individual cells. And this one allows the sort of the enzymes to go in there and be little microscopic molecular workers. So it's basically a combination of enzymes that the enzyme uses a piece of DNA as a guide to know where to act, and then it cuts specifically in response to that. There are videos, and we don't really have time to go through these now, but videos about CRISPR, video about gene editing that some students told me they found pretty helpful before. And exciting new possibilities, again, kind of scary. So here's one opportunity that gets talked about a lot, this idea that we can genetically engineer organisms that we don't like. So mosquitoes are annoying for us in other parts of the world. They bring deadly disease. So mosquitoes are the vector that brings malaria parasites. And they talk about genetically engineering mosquitoes to fight back against malaria, or genetically engineering. I've heard of like this idea we might just make mosquitoes extinct. And the technology exists, as I'll show you, where you can genetically alter one organism and then have it sort of affect future generations. So this is a technology or a concept known as gene drive. And if you take one of these genetically modified organisms and introduce it into a population, you can give it the tools to alter not only its DNA, but DNA that it comes in contact with. So normally, let's say you alter one individual, that can pass that altered DNA on like an allele. And that's not very effective in a population, right? So unless there's some kind of selective advantage, it wouldn't really go spreading through the population. And if you're trying to, say, kill a mosquito, that's not really a selective advantage for mosquitoes. Hard to breed when you're dead. But if you take this gene drive situation, then the children of affected mosquitoes, basically they're always homozygous for this effect, and they pass it on, and they become homozygous into their kids. So they pass it on as a heterozygous, but because of the gene drive, those individuals become homozygous, and then they're guaranteed to pass it on to all of their kids. And the effect there is that it spreads through the population, and eventually through this preferential passing on, could take over a whole population. So you could add this to your mosquitoes and cause them to be, let's say, I'm trying to think if sterile would help, but certainly maybe toxic to malaria parasites or something like that would be a technology that could be used here. another interesting thing about biotechnology is it's becoming cheaper. And this like CRISPR technology it's out there, it's available. People like yourselves are probably not too far away from understanding how it works and maybe being able to use it. You order up enzymes, you design pieces of DNA to make this do what you want. Kind of a scary world now where people outside of scientific laboratories can do some genetic work on themselves. So for thousands of dollars, a few $1,000s, you could get yourself do some gene therapy on yourself. And some people made news about this. This is one, why I injected myself, and then one from the next year, why I regret altering my DNA. So I think maybe by this point in genetics, you appreciate that it's a big black box in there. There's a lot of genes that have a lot of functions. We can know a lot about what the functions do, but we don't always know every possible function. So a person trying to alter their genetics, they might have a target in mind or a specific outcome in mind, and they might miss some side effect. And I think genetic manipulation is really ripe for side effects, unforeseen side effects. So I don't know about you. I will not be opting in for any gene changes to my body anytime soon. And maybe this trend will disappear, maybe, who knows. It's pretty scary. It's a whole field of like bioethics that relates to asking these questions about whether we should be doing these things, what kind of protections need to be in place, and maybe like legal protections or prohibitions on how people get this technology if it potentially could fall in the hands of people who don't know what they're getting into. All right, we kind of run out of time. I had a little bit in here about genomics. Maybe I'll talk about this in a little bit in a future class. But for now, I think I'll probably call it there. Have a wonderful Thanksgiving, and we'll see you back in the same format next week. I have an assignment scheduled to be due on Thursday next week. I don't think we'll probably be on track for that. Maybe I'll condense down the assignments, or maybe I'll just make them so darn easy, like a quiz, like I think assignment 5 was a pretty easy self-done quiz. So I'll keep you informed. I'll let you know what I come up with. Hopefully get caught up on some grading and give you a good grade update by next week. Questions? Thoughts? All right. Well, good to see everybody. Have a good week. Happy Thanksgiving.
Bye. Happy Thanksgiving.
Happy Thanksgiving.